Okay, welcome everyone. Can you hear me? I think we should get started. It's time. So welcome everyone to this symposium. In honor of mid-career prize attributed to Mike Anderson. My name is Pierre Gagnepain, and as Mike past postdoc, it's quite an honor for me to chair this symposium. I would like to apologize for my rusty English. So my first task when I was to find the main theme and topic of this symposium, was to find what this topic should be, what this symposium should be about, and then realized that there was not a single article wrote by Mike in which the word forgetting did not occur in the abstract or the keywords. So on the basis of this observation, I've decided that perhaps it was a good idea to articulate this symposium around the concept of forgetting to showcase Mike's work. As you will realize, listening to the speakers today, rarely a scientist has contributed as much as he did to the development and understanding of a scientific concept. Forgetting has long been viewed as the result of passive processes resulting from either the absence of memory reactivation, that is memory decay, or from the interference caused by other similar memories. Mike's research has focused on the idea that forgetting can also reflect an active mechanisms that depends on inhibitory control processes that are engaged to limit access to memory traces. To understand the influence of inhibitory control mechanisms over memories, Mike has developed two extremely influential and popular psychological paradigms. The retrieval-induced forgetting paradigm, which considers that selecting memories amongst competitors during retrieval engage in inhibitory control, and the TNT paradigm, which relies on the idea that inhibitory control is engaged and motivated by the desire to limit the conscious awareness towards unwanted memories. His work has a broad range of application across various domains, including philosophy, psychology, neuroscience, neurobiology, and psychiatry. And his ideas that we will try to feature today in this symposium have clearly been a source of inspiration for many of us. Today, we are extremely fortunate to start this symposium with his mentors, Elizabeth and Bob Bjorks, who will enlighten us, as you will see, on the origin of the concept of RIF. Elizabeth and Bob are the head of the Bjork Learning and Forgetting Lab at UCLA. Elizabeth is a professor of psychology and senior vice chair in the psychology department of the UCLA University. And she has also as chair UCLA Academic Senate and received the UCLA Distinguished Teaching Award. Bob is a distinguished research professor in the Department of Psychology at the UCLA department. And Elizabeth and Bob have received many awards, I cannot list all of them, and distinction through their amazing career, among which the James McKean-Cattell Award for outstanding contribution to applied psychological research and the Montoring Award for association from the Association for Psychological Science. And I should mention that there is two so the talk of Elizabeth and Bob is pre-recorded, so due to the time differences, they can't be with us. So if you have any question, we can, at the end, we can pass on to them the question you have that you can leave in the Q&A box. And there is also a chat box for informal comments during the talk.
Elizabeth and I are delighted to be taking part in this symposium in honor of Michael Anderson. We remember well his years at UCLA as a graduate student, and we're going to do a bit of reminiscing about that before we come to be a little bit more up to date. In particular, we want to reminisce about his role in our Friday morning CogFog meetings, a meeting that's now in its 42nd year. And Mike's CogFog era the early 1990s, was very active, and many of the then grads and postdocs have gone on to be influential researchers. Mike was famous for arriving a bit late for CogFog meetings until the year that Elizabeth and I were away on sabbatical, during which Mike played a critical role in keeping the CogFog meetings going and insisted that everybody show up on time. When visiting Michael's lab, we were delighted that he has created a kind of Cambridge CogFog meeting, also against his students' objections on Friday mornings. We can't resist showing a few photographs from Mike's UCLA era.
As Bob just mentioned, the CogFog meetings were very active, very lively when Mike was there. A lot of interesting discussions and arguments amongst the students, as demonstrated by this one going on between John Shaw, now at Lafayette University, and Mike Anderson. Somebody, when they left, who had visited CogFog, left us with the thought that the presenter should have a gavel to deal with such matters. And here you see John Shaw using it to get to settle the point with Mike. We also went on a lot of CogFog trips to the baseball games, to the
Horse racing
ones, to the bowl. And this is one taken on one of our trips to Disneyland. And as you can see, there's Mike. And if you look around, you probably notice that a lot of other people who've now gone on to have very illustrious careers. That's Anthony Wagner, just to the left of Mike. There's also Bobby Spellman down sort of at the bottom of the stairs with Nancy Canwisher.
Many others.
And this also shows that the CogFog people knew how to party once they were away from UCLA and its economics. And here's a number of us as a party. That's Beth Loftus and Bob in the middle there. And this particular party took place at the meeting when we presented with Mike the very first retrieval induced forgetting study. It was scheduled for 8 in the morning, which was not the most, so I didn't think we'd have a very big audience, but Mike spent a lot of time going around creating business for our talk. I'm not all sure what he promised, but on the way to that talk, we happened to run into Rich Schifrin on his way to the talk. And he looked at us over a cup of coffee with very bleary eyes and said, "This better be a damn good talk." Here's a picture of Mike graduating. And again, you can see Bobby Spellman in the picture and John Shaw. And that is also Aaron Benjamin there, all dressed up for graduation day. And
he's now editor of JEP,
right.
Learning Memory.
And was also at one time chairman of Psychonomics. Here's Mike with a lot of other very distinguished psychologists, Mike Boughton, Dan Wegner, Martin Conway, who all participated in the presidential symposium when Bob was president of Western Psychological Association.
Okay, so that's quite a bit of memory and some personal things. We'd like to also remember Michael's research contributions during his time at UCLA. And this was a period when we were formulating a theoretical framework that's guided a lot of our research, which we called our new theory of misuse. And a key assumption is that when an item in memory associated with a given cue becomes accessible in future, and competing items associated with the same cue become less accessible, but become retrieved via a given cue, other things associated with the cue become less accessible. So using our memory, we argue, shapes our memory by making what we recall more recallable in the future and competing items less recallable. Mike, though, noticed that research on the so-called fan effect suggests that instead that activation of a given cue target associative string spreads activation to non-target items sharing the same relationship with the target. So, for example, a cue such as citrus fruit : OR with missing letters might activate not only orange, but also, if less strongly, lemon. So in contrast to what we were assuming in this theoretical framework, you should get activation rather than suppression of competing responses. And so Mike, typical of Mike, designed a retrieval practice paradigm that let us look at this issue, and consistent with retrieval-induced forgetting and inconsistent spreading activation, untested items from practice categories were indeed recalled more poorly than were corresponding items from practice categories. I'll just try to illustrate that with these little diagrams that actually Michael invented at one point. But first, I need to give a bit more background with respect to the assumptions of our new theory of disuse. And one argument is that there's a very interesting kind of symbiosis of learning, memory, and forgetting. And we try to capture that in our so-called new theory of disuse. And point number one was to capture theoretically some important peculiarities of the human memory system. And one is that a remarkable capacity for storing information is coupled with a highly fallible retrieval process, that what's accessible in memory is heavily dependent on the current environmental, interpersonal, emotional, body state cues. Retrieving information from memory is a dynamic process that alters the subsequent state of the system. What you recall becomes more recallable, things in competition with it, less recallable. Access to competing memory representations regresses towards the earlier learned representation over time. And importantly, conditions that produce forgetting often enhance learning. And I just picked the story up a bit at that point. So among those conditions that produce forgetting are context variation. If you change from a context from where something was learned to some other context, there will be, that will induce forgetting, loss of access. The spacing effect itself is a dramatic illustration of conditions that produce forgetting can enhance learning. There are others as well, but let me just quickly illustrate those two. In work we did a long time ago at the University of Michigan, participants studied materials in a particular room. Then 3 hours later were tested back in that room or tested in a different room on the Michigan campus. And no surprise, they recalled more when they were back where they had learned the material than in a new place. But in the key other condition, they restudied the material rather than be tested, either back in the same place 3 hours later or in a different place 3 hours later. And then after an additional 3 hours, they went to a neutral place called room C in this diagram and were asked to recall the material. And at that point, there was a substantial advantage for having studied the material in two different locations. This has always been of interest because students can pick up a study guide at most universities that will tell them to find one good place to study and do all of their studying in that place. The spacing effect, of course, dates back over 100 years. And what it refers to is shown in the bottom pair of lines, is that when something is presented or studied twice with spacing between them, later recall, long-term recall is better. But if you just consider that at the time of the second presentation, more forgettings happened in the spaced condition, that means that this is another case where forgetting rather than undoing learning actually enhances learning. And then as far as conjectures as to why forgetting enables learning, this would be a long talk, that it might induce encoding variability, make retrieval more of a learning event. A series of different ideas have been advanced why forgetting enables learning. But for present purposes, we want to focus on, in the theoretical framework, the new theory of disuse. It represents the presumed interaction of storage strength and retrieval strength. So in our framework, items in memory are indexed by two different strengths. One, a storage strength, the degree of learning or how entrenched or interassociated something is with other things in your memory. A retrieval strength, the current ease of access or how primed or activated the item's representation is at a given point in time in a given place. Importantly, retrieval strength completely determines an item's recall probability, that is, its performance. Excuse me. We call it a framework. Elizabeth's going to get me some water. We call it a framework, but it's not a process model. Instead, it's designed to capture certain dynamics of human memory. And the implications, though, of this framework have been broad and surprising. For example, in Google Scholar searches, the combination new theory of education, there's been 849 articles, according to Google. New theory of disuse and skills, 749. New theory of disuse, teaching or training, 740 articles, and so on. And with a major emphasis is that if current performance, retrieval strength, is used to index learning, we become prone to overestimating the degree to which learning has been achieved, because retrieval strength is heavily influenced by recency and cues that are available now, but unlikely to be available later. And instructors become susceptible to choosing poor conditioned learning over better conditions, given that students on the short term will do better often with the poorer conditions. Learners themselves become susceptible to preferring poorer conditions over better conditions. Now, this storage strength, retrieval strength idea is hardly new. Estes in 55 points out that all the current learning theories are distinguishing between something like habit strength and response strength, and that corresponds to things like reflex reserve and reflex strength in Skinner's case. Even Ebbinghaus distinguished between what he calls trace strength and trace readiness. So from the very beginnings of research on memory, that distinction has been recognized. Now, it was so common because there's old evidence of latent learning, overlearning studies. There can be learning without performance. There's newer evidence, though, particularly relevant to education, that there can be performance with little or no learning. The bottom line is what we can observe is performance, what we must infer is learning, and the former is an unreliable guide to the latter. Now, in our new theory of disuse,
shaking table.
Elizabeth points out I'm shaking the table. Information memory, no matter how overlearned, becomes inaccessible a long enough period of disuse. The street address, phone numbers, friends' names, things you had for years, thought you would never forget, will become inaccessible to recall over a long enough period of disuse. But that information remains in memory as measured by recognition, priming, and especially relearning. And that's in contrast with the original law of disuse, which said that memories that are unused fade away like footprints in the sand. Thorndike was not all wrong, however, because he did emphasize that use matters and shapes our memory. And that is why we ended up calling it a new theory of disuse. The assumptions in terms of what is new is how storage strength and retrieval strength interact. And storage strength grows as a pure accumulation process. That is, you just keep building up storage strength. As far as evidence that's available, it looks like that's permanent. It's a negatively accelerated function of current storage strength. The more strength you have, the less you can gain by study, and retrieval strength. And this is the one key thing that's unintuitive about it. The higher something is in retrieval strength right now, the smaller the increments in storage strength, the smaller the learning. So letting some time lapse, the context change, all those things reduce retrieval strength and enhance learning, that is storage strength. Whereas there's no limit on storage capacity, there is a limit on retrieval capacity, the number of items that are retrievable in response to a retrieval queue or set of queues. And these are some details of the interaction that I don't need to go into. Retrieving an item and studying an item both increment retrieval strength and storage strength. But retrieving successfully is the more potent event. Increments in retrieval strength are a decreasing function of the item's current retrieval strength, an increasing function of the item's current storage strength. And decrements are higher the item's current retrieval strength, lower the item's current storage strength. Schematically, if we have items that are high or low, the four combinations of being high and low on both of these, then examples would be, for example, that childhood phone number is low in current retrieval strength, but high in storage strength if it was a phone number that was had for a long time during one's childhood. A current phone number that you've been using is high both in retrieval strength and storage strength, whereas in other cases, something like a hotel room number from some meeting last year would be low in both retrieval strength and storage strength, whereas at a meeting, the current hotel room number would be high in retrieval strength after a day or so, but low in storage strength and quickly forgotten after the meeting. So effects of a study trial, this just represents the assumptions I mentioned before in terms of particularly the kind of asymmetry between the effects of retrieval strength on storage strength. And this is effect on loss of retrieval strength, you don't lose storage strength. Now, the new theory disuse is consistent with a variety of effects, and to some degree was designed to be consistent with those effects. So that's not such a major effect. Just to illustrate one, the interaction of spacing interval and retention interval, we know that if something is studied twice, indicated by P1 and P2, presented once, presented again, then at a long delay, there's a big advantage for spacing. But at a short delay, it tends to reverse. Mass practice can look good at a very short delay, as a lot of us who crammed for exams as undergraduates might have noticed and taken advantage of. But long term, it's a very bad strategy to cram or to have massing, because forgetting is very rapid thereafter. Now, the interplay that's represented here is adaptive, because forgetting is important and loss of access is preferable to erasure. Non-recallable information is non-interfering, but it remains in memory should it be needed again, learned rapidly. Using our memory shapes our memories. What we're accessing becomes more accessible, competing information becomes less accessible, and what is most needed tends to be most accessible in terms of need statistics. What we need in the near future, typically from the recent past, the conditions that lead to disuse of recent information are often those where earlier information and skills are again relevant. So now, into this picture, we, as promised, return to retrieval-induced forgetting, this work that was triggered by Michael Anderson's observations and his initial research at UCLA.
And challenged to the new theory.
And challenged to the new theory disuse. So we already kind of mentioned using one memory shapes one memory, information retorted becomes more recallable, other information less. And in the retrieval practice paradigm that Michael came up with, participants studied a number of exemplars of different categories. So for example, they studied fruit as a category, and they studied 6 exemplars of each of 8 categories. And then a retrieval practice phase where they would see a cue like fruit OR, and have to retrieve it. And they did this for half of the pairs, 3 times each. Then a retention interval, and on a final test, were given a cue like fruit. There were different ways of doing this, and asked to remember the pairs they'd studied. And so, for example, this just illustrates again that if fruits and drinks were categories, in the fruit category, you might have practiced orange, banana, and cantaloupe, but not lemon, nectarine, and pineapple. In the other category, you wouldn't have practiced any of those. And so in terms of the practice category, fruit, you would have practiced orange. And now the question is, what's the effect on lemon, an unpracticed item? And what we saw quite dramatically was that the practiced item, orange in the fruit category, is recalled at a high level. In the unpracticed category, scotch and gin in the initial study, 56%. Then the key thing is what happens to lemon. If there's spreading activation, lemon should be higher than 56. But in this and many other experiments, significantly lower. And so the practice category enjoys a boost of some 25%, but the unpracticed members, a loss of about 15. So in the inhibitory account, items were actively inhibited during the retrieval of those. And to retrieve orange in the presence of such competition, lemon must be selected against, leading to inhibition. And in the non-inhibitory account, it's just the notion that I'm making one exemplar very available, it blocks access to others. But at this point, strong competitors, which are most in need of suppression, actually suffer the most retrieval-induced forgetting, as Michael showed, whereas the blocking interpretation predicts that weak exemplars are most susceptible to being blocked. Even impossible retrieval practice, as when letter cues like fruit ZE dash do not result in exemplar being retrieved, you still get retrieval-induced forgetting, as Benjamin Storm showed in 2006. So if you get a cue fruit LE and you recall lemon, that impairs later recall of apple and pear. But if I get a fruit ZE and nothing comes to mind, I still have to select against apple and pear, even though I'm not creating any specific competitor. And when you do that, you find that impossible retrieval practice causes as much inhibition as actual retrieval practice. So retrieval-induced forgetting in the real world is the ability to recall family abuse incidents from one's past. It's relevant to that. Slowed access to one's first language at time of the second. But how about effects of practice exams? Is this where you're finally going to pitch in?
Right. So we often give practice tests, at least I did for many years in my courses, and I always thought that they would be a benefit of students. But it's possible that because when we give practice tests, often which are multiple choice tests, we're asking our students to select one answer out of, say, 4 alternatives that they think is correct and reject the 3 others. So maybe we're creating retrieval-induced forgetting for those 3 incorrect alternatives, although they may be answers to other questions of importance about material in our class. So that was a question we wondered about. Would multiple choice practice tests be, with multiple choice practice tests, are we actually doing a disservice to our students by creating retrieval-induced forgetting for the incorrect alternatives? And I'm not going to go into the details of the experiment, but we looked at this carefully in an experiment and where we compared competitive multiple choice tests to cued recall practice tests. And what we found, as you can see here, is you get an advantage for previously tested questions. So as to be expected, if a question appeared on a practice test and now it's appearing again later on the final exam, it has an advantage over things that weren't ever tested before. But what about tests that you might call related questions? Questions for which one of those rejected alternatives is now the correct answer. How do they fare? Do they suffer from retrieval-induced forgetting? And what we have shown here is that that is not the case with the multiple choice tests, although it is the case with the cued recall test.
So we've summarized some of the things that were related to the impact Michael Anderson had on us during his short time at UCLA. And now, when Mike's being honored for his work, it's exciting to look back on these things, but it's also very impressive that when we look at Mike's accomplishments, what we have summarized was an important beginning, but only a beginning. What Michael has done, both in terms of brain mechanisms and in terms of inventing paradigms like the think, no think paradigm, is really verges on amazing. And he's very deserving of this award. And we from across the ocean are delighted to join in all of you who are there in congratulating Mike on this achievement. And with that, that's the end. I don't know. We weren't very good, but maybe we could, if we can figure out how, we might send them this one and say we're going to try to create an improved version.
Yeah.
Now you think you know where this went, right?
Well, what did we call it again? Okay, so let's see what this says.
Welcome back, everyone. That was an absolutely fascinating talk. That was really fantastic to learn about all this concept. So unfortunately, due to the time difference, Elizabeth and Bob cannot be with us. So if you want to leave any question for them in the box, we'll be happy to pass it on to them. So now it is my great pleasure to introduce the next speaker, my dear friend Maria Wamberg. Maria was, after her first postdoc at the University of Magdeburg with Alan Richardson-Claven, Maria moved to Cambridge at the MRC-CBU, where she worked with Mike, before moving to Birmingham as a lecturer, senior lecturer. Then after a promotion to reader, she finally moved now to as a full professor to the School of Psychology and Neuroscience at the University of Glasgow. And welcome, Maria.
Hello, everyone. Let me get my screen up. Can you see that? Yeah. Okay, yeah, really nice to be here. Although, of course, it's a shame we can't get together and celebrate this occasion in person, which I would have liked very much. And thanks a lot, Pierre, also for putting this together. And let me start by finding Mike's face here in crowd. there he is, looking very pleased to have gotten this award. I really want to start with saying a big, huge and warm congratulations to you, Mike. It's very nice to see your work recognized in this way. And of course, we all know the massive influence that you've had on the field, you know, not just the field of experimental psychology, but also forensic psychology, clinical fields, and so on. So it's very nice to sit here and be able to talk in this symposium and say congratulations. Also, thanks for the mentorship and all the lots of inspirations that you've given over my career, especially in my younger years. And, you know, I'll take the silly picture off the screen in a second. But I just want to say, maybe very briefly, I'm not sure if we ever talked about these beginnings, but obviously Mike Anderson had a very strong influence on my career, or his works have had a very strong influence on my career. And when I started doing a PhD, or when I planned for a PhD, I wasn't the least bit interested in episodic memory at all. I was, like many people, I think, in the early 2000s, really into cognitive control, attentional gains. How does the brain do that to enhance and target information and suppress distracting information in the environment? And I was particularly interested in this phenomenon of negative priming. So if you, or, you know, this was for me a nice task to demonstrate the strong effects of attentional gains and losses. So if you do a task here where you have two hybrid images, you only have to respond to one, but there's a second interfering image. So in this case, you'd have to respond to the red images. So you'd have to respond to the chair, but there is a duck in the background. If this duck now becomes the target on the trial n+1, so on the next trial or one of the future trials, you're slower or worse at responding, making a correct response to this thing that had been ignored on a previous trial. And so this was something I was really interested in, and I wanted to do a PhD on this topic. Unfortunately, none of the supervisors in the attention field wanted me as a PhD student. And the one supervisor who actually offered me a position wasn't the least bit interested in attention, but he said, well, you know, you should go and read Anderson, Bjork, and Bjork, because really there is a phenomenon in episodic memory called retrieval-induced forgetting that is not so much unlike this negative priming effect. So, you know, as Elizabeth and Bob just pointed out really nicely in their talk, if you have a memory cue that is linked to two competing images, and your task is to only retrieve one of these two images, what I call the target here, then this target gets a boost from a lot of information flowing into this target, but you also see a suppressive effect, like a negative boost, a loss for every competing item that's co-activated by the cue. And you can see that in behavior. So if on trial n+1 or at some point in the future you test memory, you see this boost that the targets that had already been retrieved are receiving, but you also see this, this impairment for items that had been unselected or ignored in previous trials. So really, this got me into episodic memory in the first place. And obviously, I've never looked back. I got hooked on the topic and fell in love with the topic, but that was the beginning of my personal scientific journey. And so then in my PhD years, I started to work a lot with the methods we had at hand at the time, with mainly univariate fMRI, trying to see what the neural underpinnings of this retrieval-induced forgetting effect were. Do we really see that this, you know, there are certain networks that are also involved in attention that are also critical for creating these suppressive after effects in memory? And in fact, we did. So I'll show you a few pictures here, but the networks that we observed in a number of studies over the years really do look a lot like what people in the attention world, John Duncan, for example, calls the multiple demand network. And in particular, so this is activation in response to a selective retrieval condition, and this tends to increase the more competitive retrieval becomes. So the more you have a need to select, the more these networks tend to increase. And that's the same phenomenon as you see in attention studies as well. And in particular, these mid and inferior prefrontal regions, the more engaged they are, the more a participant tends to show the suppressive after effect. So we observed this relationship to behavior in a very first study I did during my PhD. We also observed that the more you engage these inferior prefrontal regions, the more you see a neural after effect when you later have to activate an item again. And then in a third study in the series here, we also showed that there is a genetic variant that influences the amount of dopamine people have in their prefrontal cortex. And this variant has been very strongly related in computational models and in previous work to attentional selection, so attentional filtering. And we showed here that people with a lot of sustained prefrontal dopamine are the ones that have the highest ability to suppress distracting memories as well, and show the strongest memory, the suppressive after effect, retrieval-induced forgetting. And they are also the ones that engage their inferior prefrontal cortices the most. So very consistently, we find these attention or previously attention-related networks, and especially inferior prefrontal activations related to retrieval-induced forgetting. However, I found just showing that these networks are engaged a bit unsatisfying. And so after, I guess in the middle of my first postdoc, I contacted Mike and asked whether he'd be interested to try and do a study in Cambridge where we try and really track the forgetting of these memories directly by observing the brain activity patterns related to those memories. Mike said yes, luckily. So we wrote up a fellowship together, and I joined his lab in Cambridge for 2 years. And this is one of the main projects we did there. So we designed pretty much, an elaborate version of a retrieval-induced forgetting paradigm, I would say, where our participants first in a learning phase had to associate relatively abstract words to one of two different stimuli. So in a first go, as a participant, I would associate this word snow to the face of Marilyn Monroe, for example, by creating a very vivid mental image where Marilyn's lying in the snow, or whatever you can come up with. And I would associate the word antique to Albert Einstein's face. But then in a second round, I have to associate a second item to each of these cue words. So now I would have to link the hat to the keyword snow as well, or the goggles to the word antique. And importantly, these always came from different categories. That all happened outside the scanner. We made sure that people learned these associations well. And then in the scanner, we would present our participants with some of these cue words they had learned before, and instructed them that they'd always have to retrieve the first image that they learned to associate with this particular word. So their task here was to retrieve Marilyn. And the assumption was, even though we never told them actively to suppress the hat, but the idea is that the hat would elicit interference and thus have to be downregulated in order to successfully retrieve the first association they had learned. Each of these cue words here, like snow, appeared 4 times overall in this reactivation or retrieval practice phase. So we could track the brain activity pattern across these 4 repetitions. And importantly, we also had these control triplets here that never actually appeared in the scanning phase. So these were just associations they had learned previously, faces, objects, and scenes floating around in the brain, but they were never actually triggered by any of the cue words. Finally, importantly, we also had a localizer, or that is a key, the key element here. We had a localizer where we just, at the very end of the scanning run, we presented people with these images they had learned initially in isolation on the screen over and over again. And from these multiple presentations, for example, of Marilyn and the hat, we could sample what in an individual participant's brain, the typical Marilyn pattern would look like, or the unique hat pattern would look like. And that then allowed us to ask, well, how much is this Marilyn pattern active when we cue them with the word snow, compared to how much is the hat pattern active or inactive? And that was all relative to, for example, the Albert or the goggle patterns that weren't triggered by any of these words. So what did we find? I'll start with the Marilyn pattern here. So the graph you see here is the 4 repetitions of the word snow triggering triggering patterns here in these inferior ventral visual stream areas. And difference R here means this is the level of correlation with the Maryland pattern that goes beyond just a normal phase pattern, right? So we see phase-like activity all the time when people are retrieving a phase, but how much are the voxels that really represent Maryland individually active on each of these repetitions? And what you see here is a moderate but significant upregulation across repetitions of this Maryland pattern, which reflects more and more successful retrieval of the individual Maryland. More interestingly for us in the study, we also observe a very strong modulation of the hat pattern, even though, remember, they're not supposed, we never explicitly cue the hat, but you see that there's very strong initial co-activation of this unique hat pattern during the first repetition, followed by a very strong and significant down-regulation of the individual voxels representing the hat down into this gray zone here, where the hat really becomes significantly deactivated below other objects, for example, that are never triggered by any of the cue words here. We also find that when we do a memory test later in the scanner, or after this whole scanning is completed, we find that if we ask people to tell us which of these two hats they initially saw during learning without referring to any of the cues, we have a significant behavioral impairment, which is just a visual form of retrieval-induced forgetting here. So they are worse at discriminating this hat from a similar lure, presumably as a consequence of having suppressed the hat all along during this previous scanning phase. So this is an after effect, a retrieval-induced forgetting effect. We also find that the size, like across participant, the size of this suppressive after effect correlates moderately but significantly with the slope, or sorry, with the degree to which people down-regulate the voxel patterns. And finally, to link to a bit to what I showed you previously, we also see these typical candidates that we always see, especially in prefrontal regions. And again, like in previous studies, the more our participants engage these mid-prefrontal regions, the steeper the suppression slope for the visual patterns in their sensory cortices. So to sum up this first part, that is really the most related to the work I've done together with Mike, or inspired by Mike, we find, like Mike and many others have in the past, that remembering selectively induces forgetting of co-activated, competing, potentially distracting memories, at least for a little while. We find that the brain does engage control mechanisms that look very much like attention control networks to adaptively down-regulate these competing memories. So there's always a relationship between the degree to which these regions are active and the degree to which they show these suppressive after effects. And we also show that you can actually track this retrieval-induced forgetting directly in the brain patterns that represent unique individual memories. So I've talked a lot about the hat so far, I guess, so that these competing memories that tend to pop up, or we think pop up, and we have to adaptively down-regulate. For the second part of my talk, I want to talk a bit more about more recent work where I've become more interested in Maryland, or in more scientific terms, in these target memories. So since leaving Mike's lab, I've really focused more on situations that don't necessarily involve competition, although I think memory retrieval always involves some degree of competition. But we started wondering about much more basic questions. How does this cue to call work at all in the brain? When I have an unrelated, just associatively linked cue, how does the brain reconstruct a previous image? What features of an image can be reconstructed at all? Which ones can we track in brain activity? When in time and where in the brain? And also when we repeatedly cue ourselves to retrieve a visual image, how does the representation of this image change with repeated remembering? So it's not too far from what we've done before, except we're focusing on the target memories that we really want to retrieve now. And so when asking, well, where in the brain and how does this cued recall process work, I started looking into the literature and figured, well, we know a lot about how information enters the brain and how it's analyzed in a bottom-up feed-forward way when the image is visually on the screen in front of you. So when seeing a chair, for example, we think that, passing through the thalamus to early visual cortex, early visual brain regions start analyzing low-level features like shapes and orientations, things like that. And then as the information travels forward along the ventral visual stream, it's sort of being put together into more coherent shapes and abstracted along the way. And it's believed that these very late ventral visual stream areas represent concepts rather than the visual features of the images themselves. So they represent the concept of a chair or a turtle. And so we thought, well, if these are the latest stages that the information passes through before being bound together by the hippocampus, it makes sense that when an image is reconstructed from memory, a process that we believe starts with the hippocampus, the brain would do this, or would maybe first back project into these high-level abstract semantic areas. And this is the level at which we first access information when we recall it from memory. And then the brain would start backwards reconstructing the lower level visual information that was associated with a certain sensory experience. So when reconstructing the chair, we'd first activate the concept of, oh, there was something with the chair, and then it would take a while, a backwards reconstruction to get to the lower level features like this yellow orangey color of the chair, for example. And so to test this idea, we constructed different stimulus sets where our stimuli, our images, always differed along two preset dimensions, one more low-level perceptual dimension and one higher-level semantic dimension. So in this example, in the first experiments, our lower-level perceptual dimension was that the image could be presented either as a photograph in color or a line drawing. And the semantic dimension was whether it was a living or a non-living thing, so an animal or an object, in fact. And the idea behind that was that, well, if a brain region, like an early brain region here, cares more about the surface level details, it should really group together all of the photographs and all of the line drawings and not care so much about the actual content. And if a brain region like these anterior ones cares more about the content, the semantics of the image, it should group animals and objects and not care so much about the presentational format in which people see these images. And so we used these in a very simple associative memory paradigm, just like I've shown you before. So our participants have to link a keyword to one of these images each. And later in a retrieval phase, they see the keyword, they have to mentally reconstruct the image and press a button as soon as they have the image back in mind. And importantly here in this first experiment, we measured EG while people saw these images either on the screen during encoding or they reconstructed them from memory. And what we did then is to use these EG patterns here in a time-resolved way to train two different kinds of classifiers. One that was supposed to distinguish based on these brain activity patterns whether an image was a line drawing or a photograph. So that was a perceptual classifier. And on the other hand, a conceptual one that was learning based on these brain activity patterns whether it was an animate or an inanimate thing people were seeing. And the idea here is that, well, if there really is a forward visual processing stream during perception, we should see this perceptual classifier peak first, followed by the more semantic one. And if this information flow is reversed during retrieval, then this pattern should also reverse, and we should see the conceptual classifier peaking before the perceptual one. What you can see is the average decoding peaks. There's more data in the paper, but that's the easiest one to show, I guess. So we do find a trend, but not significant, for the perceptual classifier peaking before the semantic one when people are seeing the image on the screen. We see a very strong pattern of reversal, so that relative to the time they press the button and say, yeah, I've got the memory back in mind, the semantic information comes back in brain activity at least about 200 milliseconds before the more perceptual information is visible. And you can also see this in very simple ERP. So if you compare, what differentiates a photo from a line drawing or from an inanimate thing, just an average brain activity, you see, the perceptual component comes before the semantic one during perception, and this reverses very strongly during retrieval. And I think this second peak here is maybe the reason why we don't get a significant difference here, but I won't go into any details given the time limitations. So what I do want to show you is that we also turned this into a behavioral paradigm that's turned out to be quite useful. So we wondered if we can see this flip or this retrieval related flip in brain activity, maybe we can also measure this just in behavioral data. And so we kind of turned our participants into pattern classifiers by asking them on each trial one of two kinds of questions, a semantic or a perceptual one. So if you're a participant here, I would present you with a question: is the object that will come up on the screen in a second a photo and a line or a line drawing? Picture comes up and you just press the button as fast as you can. And we thought, well, if we can see a forward stream, people should be faster at responding to the perceptual features than the semantic ones when they see them on the screen. But then we also do the same task in a memory group that had linked words with these images before. And we thought, well, okay, if these images are reconstructed in the reverse order to perception, we should see a reversal in the reaction time patterns as well. So when retrieving from memory, they should now be faster retrieving the semantic than the perceptual attribute. And this is what we saw in the first experiment here, very strong effect where during perception, people are indeed faster at answering the perceptual than the semantic questions, and that flips around for memory retrieval in almost every single participant. And this is a phenomenon that now over the last 3 or 4 years, we've replicated, I think, 10 times overall. It's really super robust. It doesn't depend on the particular dimensions that you manipulate here. So across these different experiments, we've shown that it's robust to variations in the types of perceptual and semantic dimensions we use, in the types of cues we use, whether we use words or scenes or locations and so on. You always see this pattern then when an image is on the screen, people judge the perceptual aspects faster than the semantic ones, and this flips around in memory. So to sum this up and get closer to the end, from this study and many others in the lab, we think we have very good evidence that really when you do a cued recall, you retrieve an image from memory that you have to actively reconstruct, information flows in the reverse order from the original perception, triggered by the hippocampus from abstract conceptual to more early visual perceptual areas. And so for the very last study I want to show you here, we asked, well, if this is the case, if you always prioritize abstract conceptual information during memory retrieval, then maybe repeated remembering induces some kind of semanticization. So if you continuously strengthen these semantic aspects by retrieving them first and with more confidence, repeated retrieval should have an effect on how memories change with repeated remembering. And so I hope you can see the small print here on the screen. In the last study I want to show you, the paradigm was pretty much the same as in all of the previous ones I've shown you. Simple cued recall task. People learn these associations between words and images, and then they recall them being cued with a word and answering one of these two types of questions, semantic or perceptual. But now they don't only do this once immediately, but they do this 6 times overall. So in 3 cycles, each cycle answering or retrieving the image with a semantic and a perceptual question. Then we send them home for a 2-day break. 48 hours later, we bring them back into the lab and we probe their memory once more with a semantic and a perceptual question, and also some paper and pencil testing. And what we see here, I know that's a lot of bars, but let's start just with the left one here. So this is just a replication of the finding I've shown you 5 times before already, that when people recall this information from memory, they are faster at responding to the semantic than the perceptual attributes. That's an effect that stays pretty much there all along on the first day. It doesn't change significantly, even though it's just about not significant at the end of day one. But when we bring people back into the lab and we ask them again to remember what they learned 2 days before, you see a massive increase in this gap between access to semantic features and access to low-level perceptual features. So they're now very slow at remembering the details and still quite fast at remembering the semantic gist, if you want. So now this could just be an effect of passage of time because 48 hours have passed, or it could be an effect of having retrieved the item actively already on the first day and thereby having strengthened these semantic attributes. And so we ran a second group, a restudy control group that did the exact same thing as our first group, learn the words, have a final test on day 2, but in between, instead of actively retrieving these associations, they just relearned them over and over again answering these semantic and perceptual questions. And like before, as we expected, when the image is on the screen here, they are faster at at judging the perceptual content than the semantic one while doing this task here. But when we bring them back into the lab 48 hours later, they actually don't show this increase, that there isn't even a significant gap between semantic and perceptual feature access. And if you zoom in just on these delayed test results, you see that active retrieval on the first day really seems to have pushed the semantics or strengthened the semantics of these associations, these images that people are retrieving, an effect that we don't see in the restudy condition. Right, so I think I'm running a bit over time already. Just summing up here to say that all of what I've shown you in this talk is very consistent with a little framework that we put forward a few years back with colleagues in Princeton, where we argued that this magic of active recall, of really actively shaping the memory landscape within which a recalled memory is embedded, we think that this is due to its imperfection, basically, because retrieval spreads activation to other information in memory. It's not as selective as vision. The spreading activation can actually help, adaptively help the system to reduce interference from all these coactive memories and induce forgetting this way, as described in the first part. But also this can help to embed a memory into a larger semantic network and lead to its stabilization and generalization over time. And with that, I'd like to thank you a lot for your attention. Thanks to all my collaborators on these studies, and especially to people in the lab, some of which have left in the meantime, but these 3 in particular were the ones involved in the studies that I've shown you. And congratulations again to you, Mike, for this very well-deserved award.
You need to stop showing your screen, I think, Maria. Yeah.
Yep.
Thank you so much. Okay, I can see that there is a question. I have a question for you, but we don't have much time, so I'm gonna ask the question that I can see in the chat. So hi Maria, great talk, thanks. This is a question from Joe Taylor.
Hi Joe.
In the behavioral paradigm, can you distinguish what type of information is reactivated first from the difficulty of the decision? I imagine perceptual information is just less relevant for people when trying to remember, so then these judgments are harder. Would there be a way to dissociate these possibilities?
Yeah, I think it's a good question in general. Could this be just a trivial effect of difficulty? I think it could be a non-trivial effect of difficulty, but it has to be specific to the conditions in which people are doing these judgment tasks, right? Because if a perceptual question was just harder to answer than the semantic one, then we should also see that people, no matter if they see the image on the screen or retrieving it from memory, would find it, would be slower at answering a certain type of question. So I don't think it can be a trivial difficulty effect, that generally the perceptual decisions are harder to make than the semantic ones, but it has to be an interaction with a process that you're in, whether you're in a sort of feed-forward visual state or a memory state. So I think it can't be completely trivial. Although it can have to, the question of whether this sits at the forefront, sort of before the decision bottleneck, or more at the decision stage, is one that we're working on at the moment.
So thank you, Joe, for your question. I have a question for you, Maria. Thank you for your fascinating talk first. I think it's a $1 million question, actually. So you showed in the RIF paper, in the Nature Neuroscience paper, you showed that there is an enhancement of prefrontal activity with competition. And I'm wondering what is the meaning of this enhancement, because it could mean two things, in my opinion. One thing could be that you enhance the memory through attention you enhance activity of the target memory, which then create lateral inhibition in the neocortex. Or the other mechanisms could be that this increased activation that you see in the prefrontal cortex could reflect some kind of direct inhibition of the competitor in the neocortex. So one mechanism would be lateral inhibition through rotational enhancement of the competitor, and the other mechanisms would be kind of direct inhibition or feed-forward inhibition, if you like, of the competitor. Is there any things in your data that can say something about these two, or in the literature? Do you have either progress on this question in the literature? Or?
Well, doesn't Pierre Gagnepain have a paper using dynamic causal modeling to try and get at these possibilities? So I can't think of anything in the RIF world. We've never tried directly assessing this. It's obviously one of the important questions. Has the prefrontal cortex really got this power to directly act on these visual representations? I think Mike has a comment on that. But I personally have never tried to dissociate that. I think both are possible. There's computational models like from Ken Norman's lab, for example, that suggest that, that there are these K-winner-take-all mechanisms that can actually act on coactive information without having a homunculus sit in the prefrontal cortex. But I think the jury is still out there also in the attention world, and I can't think of a definitely, distinguishing study in the memory field.
Okay. Thank you so much. Okay, so I think now it's time for the tea break, and we shall come back in half an hour for Roland Benoit's talk. So I'll see you later, guys. Thank you so much. Thank you, Maria.
Bye.
Thanks, Maria. Thanks, Pierre.
Sorry, Pierre, are we staying online here as panelists, or do we?
I don't know. I think
I'll just mute and
you can turn your video off and mute yourself, and then come back in, and it just means that you'll stay on this side. Yeah.
But are we on the public side?
Right. When your video is on, yes.
Okay.
Yeah.
Okay.
Okay.
Hey, everyone.
Hey, Roland, I think this is
Roland.
Hey, I think you are public.
You are public.
We're all public.
We are
Okay, so
public.
I have to behave now.
Yes.
Can I check? Are you Roland?
That's me, yeah. Shall
Yes. Okay.
Oh,
You
I
can't
don't.
still as Pierre. You can
Oh, yeah.
change your name if you go to participants, and then there's a more box, and there's an option to rename.
Yep.
So you can change
Yeah.
your name. And we'll be starting. We've got 5 minutes.
Let me see if I can share my screen. Could you be so nice to confirm what you see?
Yeah,
Yes.
I can see your full screen.
Yes, we can see your presentation, Roland.
With all the smart, without all the smart remarks that I'm going to make, right?
Yes. Without it.
All right.
Perfect.
Yeah.
You're welcome to keep it up. We've got 5 minutes, so you can also turn off your video and mute yourself until Pierre will come back in when it's time to start.
Sounds
And
great.
then you can come back. Okay.
Thanks for organizing this. Thank you.
See you in 5 minutes.
Yep.
Great. Okay, welcome back, everyone. I hope that you can hear me. It is now my great pleasure to introduce Roland Bonnat. Roland have done postdoc with Mike at the MRCCB in Cambridge, before then moving to Harvard University with Dan Schachter, where he stayed there as a postdoc until 2016, before moving now to the Max Planck research group, the Max Planck Institute, as a research group leader in Leipzig, Germany. So in the Human Cognitive and Brain Science unit. So thank you so much, Roland, for being with us today. And it's all yours.
Oops. Well, thanks, Pierre. It's my great pleasure, obviously, to contribute to this symposium in honor of Mike. We've already heard quite a bit about Mike's groundbreaking work on retrieval-induced forgetting. And that work, of course, has formed our understanding of forgetting as the byproduct of an active process. Another seminal contribution of Mike is the demonstration that forgetting cannot just arise as such a byproduct. Instead, it can directly be induced in an intentional manner. And today I will talk about some of my work on such memory suppression. And this work, of course, originated from my postdoc with Mike and now continues in my lab here at the Max Planck. So what do I mean by suppression? And let's start with an example. So what do these boots remind you of? Maybe it's a stroll through the countryside or a recent visit to the beach. At least in some parts of West Germany, these days they actually may serve as a strong reminder to a recent fairly traumatic event. There's been this extreme flood disaster that caused huge damage to several communities. The boots may have been just this incidental detail of this initial aversive experience, but nonetheless now they can actually act as a strong reminder that triggers the involuntary retrieval of such memories. Now, 2 decades ago, Mike has started a rich line of research that has indicated that we are not at the mercy of such intrusive memories. Instead, we can control them by actively preventing their retrieval. So to study such memory suppression in this paper, this Anderson and Green paper, they developed the no-think procedure. And here I will show you a variant of the procedure that we developed back in Cambridge with Charlotte Cooper as the lead. So here in an initial step, we have participants associate neutral objects with aversive scenes. And much like the boots, these objects also feature in the respective scene. And we ensure that they can easily retrieve the scene whenever prompted with the object. Participants then enter the critical suppression phase. Here we show some of the reminders repeatedly with a green frame, and the task is simply to recall the associated scene as vividly as possible. And we present another third of the reminders with a red frame. These are the suppression trials. So here people's task is to keep the unwanted memory out of mind, and if it does intrude into their awareness, they are supposed to push it out. And notably, you notice that there are a third of the items that we not present at all during this period. So these are our baseline items that serve to assess the degree to which forgetting just occurs with a simple passage of time. Now, after this task, we finally assess people's memory for the scenes. So here in this task, we show them the reminders again, this time with a neutral frame, and ask them to describe the associated scene in as much detail as possible. And this is then a typical result pattern. This could be quite a number of different memory measures. But what we typically find is that people are worse at remembering the suppressed memories than at recalling the baseline memories. Right? And this difference is what we call suppression-induced forgetting. Right? And it indicates that suppression causes forgetting that essentially exceeds the rate of forgetting that occurs for the baseline items. However, what mechanisms may be at play that transiently prevent unwanted retrieval and that eventually cause sustained forgetting? Or in other words, what is it that you could do to prevent a memory from coming to mind when you're confronted with a potent reminder? those early studies were fairly agnostic with respect to the exact mechanism that people engaged for preventing retrieval. However, over the last decade, we've provided evidence that suppression can be achieved by two distinct mechanisms, one which we refer to as retrieval suppression and the other one thought substitution. So retrieval suppression, so that's the first mechanism, and it essentially prevents a memory from coming to mind by inhibiting the retrieval process. And retrieval suppression is typically associated with increased activation in the right dorsolateral prefrontal cortex and reduced activation in bilateral hippocampus. And this indeed is a pattern that Mike had already described in a seminal paper in 2004. And they interpreted the pattern as reflecting a systemic inhibition of hippocampal retrieval processes that originate from the right dorsolateral prefrontal cortex. And later on in this paper, we essentially made the case that this pattern really is tied to this mechanism of retrieval suppression. And in that study and in several others since, we have also used dynamic causal modeling to establish that the right dorsolateral prefrontal cortex indeed exerts a pronounced top-down inhibition of the hippocampus. Okay, so much about retrieval suppression. What about thought substitution? So thought substitution is not based at all on a systemic inhibition of hippocampal retrieval, quite the opposite. The idea is that thought substitution redirects the retrieval process towards the recollection of an alternative, a substitute memory. And this memory then occupies the limited focus of awareness and thus prevents the unwanted memory from coming to mind. And using fMRI and connectivity analyses, we have shown that thought substitution engages control processes that are mediated by the left ventrolateral prefrontal cortex. The idea is that these processes then facilitate the selection of the presumably weaker substitute memory. And I guess after the earlier talks, particularly also by Maria, you will share the intuition that thought substitution has much in common with retrieval-induced forgetting, so may essentially be maybe an intentional version of getting at the same effect. All right, so there are two mechanisms that seem to work in opposite fashion, one by shutting down retrieval and the other by using the retrieval process for the recollection of a substitute memory. And notably, we and others have provided evidence that both of these mechanisms also have sustained after effects. That is, they can cause forgetting of the avoided memories. However, you may wonder how big this effect is and whether it actually constitutes a beneficial mechanism to cope with intrusive thoughts. Now, particularly the second one, if that's the case, we expect suppression to be deficient in people, for example, with anxiety and depression, people who also experience quite a lot of intrusive thoughts and may therefore not be, and that may be a signal that they are not efficient in exerting suppression. Now this fellow here is Davides Ramaja, a former awesome postdoc in my lab. And to answer both of these questions, Davide led a meta-analysis in which we surveyed the studies that included both a new topical control group as well as a clinical population. And the idea is that the healthy group essentially allows us to get a sense of the effect size obtained in those studies, and that those clinical populations help us to address the second question, right? So it's therefore just also on a subset of the rich literature on think, no, think, right? So essentially for this meta-analysis, we did a quite extensive literature search for studies that had used the think, no, think procedure, and that also included one of several disorder-related terms such as anxiety, repression, depression, and stuff like this. And we used standard databases, but also looked for unpublished master and PhD theses. And in addition, we contacted, I believe, 56 authors who had previously published think, no, think studies to also get as much unpublished work as possible into this analysis. And then of those 240 studies, again, we essentially included studies that had compared a healthy sample with a clinical sample on the think, no, think task. Yeah, we did not just include clinical samples, there were also some subclinical samples. So these were studies where participants were selected based on pertinent features, for example, that they scored particularly high on the Beck's depression inventory. So this, in the end, left us with 25 studies. Each of those included at least one subclinical sample and a healthy sample, and sometimes actually various correlated measures of suppression-induced forgetting. So for our analysis, we essentially used a 3-level random effects model to account for the hierarchical dependencies of the effect sizes, and to account for the correlated effects, we estimated robust confidence intervals. And what you see here is actually the forest plots of the healthy samples, right? So this is the standardized mean change, so essentially like HSG. So a greater number indicates greater suppression-induced forgetting. And in our model, we observed that overall the effect size was medium to small, but it's important to note that the studies were quite heterogeneous with respect to the instructed suppression mechanisms. So some studies had people figure out by themselves how to achieve suppression, while others instructed for thought substitution or retrieval suppression. And in a moderator analysis, we indeed observed that the exact effect size did quite a bit vary with the nature of the instructions. So you see that it was substantially larger when people were instructed for retrieval suppression or substitution in the study. So people are better at suppression when they exactly know how to accomplish this, in a sense, right? Okay, so what about the clinical populations? So these are essentially the studies clustered by sort of symptomatology. So this is the anxiety cluster with anxiety disorders. It also includes 2 studies on PTSD, even though they are technically not considered anxiety disorders anymore, I suppose, according to the latest DSM. Then we have a depression cluster. We have a cluster of studies of people who, according to their self-reports, are high repressive copers, so who engage in a repressive coping style. And finally, we have a mixed cluster. This is really just sort of a collection of very heterogeneous studies that it's really hard to make any sense of, but that we couldn't cluster in any other meaningful way. Now, down here, you essentially see the estimates of the meta-analytical model for the healthy groups, the anxiety group, the depression group, the repressive copers, well, and the average, and the mixed one, of course. Now, the notable thing is that, as predicted, the effect sizes for the anxiety group and the depression group were actually smaller than the one for the healthy control groups. And by themselves, they were not significant in any group. In fact, quite interesting, if you look at the anxiety group, if anything, that group actually showed sort of a better retention for memories that they had tried to suppress. Okay, so these are the results. Of course, meta-analyses are very susceptible for publication bias, so that's a huge issue, right? And this is probably also the case for this meta-analysis, despite our attempts to include unpublished studies. And we also did run some metrics, which actually, like Egger's regression test, which did not provide evidence for publication bias. However, for what it's worth, we obviously also tried to adjust for publication bias, for example, via the fill and trim procedure, which essentially resulted in an almost identical pattern. So yeah, so therefore, essentially to sum up, I hope to have shown you some evidence here that memory suppression, at least in those studies, has at least a small to medium effect size, but that it can be substantially larger when people directly know how to engage a mechanism that we know should work. I've shown you some evidence that this is deficient in individuals with anxiety or depression, which we take to suggest that deficiency in suppression may constitute a predisposition for disorders characterized by intrusive thoughts. And of course, Pierre's talk later in the symposium will focus on this and actually, in fact, dwarf some of the conclusions that we can make with this meta-analysis. Yeah, I've mentioned this. It seems to be robust to adjustments for publication bias and also to outliers. So then in the next part, I want to quickly talk about another study in which we want to understand the newer basis of suppression-induced forgetting. So essentially, by now, we've made quite some progress in understanding the newer mechanisms that transiently prevent retrieval, right? So this shutting down of the hippocampus, for example. However, what is the newer basis of the sustained after effects of suppression? And Ann-Christine Meyer, a fantastic PhD student in my lab, addressed this question. And the idea is the following. Obviously, when we retrieve a memory, this is thought to be based on the reactivation of the memory's neural representation. And we hypothesize that suppression essentially deteriorates these representations and thus causes a sustained reduction in the potential of these representations to get reactivated. Now, how do we test this? So to examine this hypothesis, we extended our procedure, so the Cooper et al. Procedure. Specifically, we've added additional tests just before and after suppression. Here we prompt participants to recall each of these scenes and to indicate the vividness of their recollection. And we expected a greater decline in scene reactivation from the pre to the post test for the suppressed memories than for our baseline memories. But let's turn actually first to the vividness data and thus to changes in the phenomenological experience induced by suppression. So we have actually used this procedure in both a behavioral study and a subsequent MRI study. And I'm here plotting the reduction in vividness, right? So larger number indicates a stronger decline. I'm showing you this for the baseline and the suppressed condition. And indeed, you can see that the decline was substantially larger in the suppressed than in the baseline condition. And we replicated this finding in the fMRI study, right? Again, with a stronger reduction in vividness for suppressed and baseline items. I think this is actually quite interesting in itself, giving quite some evidence that subjective measures of memory and objective measures of accuracy can actually dissociate, right? But here we actually show that suppression also affects the phenomenological experience from memories per se. Now, we had hypothesized that this reduction is based on a sustained reduction of the memory's potential to get reactivated. And to address this question, let's turn to the new imaging data. So essentially, to examine scene reactivation, we had participants watch a series of aversive scenes, similar to the ones from the actual task, and they also watched a stream of morphed versions of those scenes. And we then trained a classifier, a support vector machine, to distinguish the activity patterns associated with the processing of these two stimulus types. And this classifier then yields a weight pattern that indicates how much each voxel is contributing to the brain state associated with processing aversive scenes. And we then calculated the dot product of this weight pattern and every trial-specific brain map, and the resulting value then essentially quantifies the estimated scene evidence on a given trial and for a given condition. And we can then examine changes in scene reactivation by subtracting the evidence on the post-test from the evidence on the pre-test. So a greater value, again, indicates a greater decline. And given the distributed nature of memory representations, we first examined this effect for a classifier that was trained on the complete gray matter. Right, so here I show you the scene evidence reduction, so pre-evidence minus post for the baseline and the suppressed condition. Again, greater indicates greater reduction. And we do find a significantly stronger reduction for the suppressed items. Now, in addition to taking this global view, we also zoomed in onto the parahippocampal cortex. Now, this region is particularly obviously involved in the processing of scenes, and activity in this region has previously been associated with the vividness with which memories for scenes can be recalled. So this is why Ann-Christine essentially took the structural scans and drew the anatomical ROIs on each participant's well, structures can. And for the left parahippocampal cortex, indeed, we observed a stronger reduction for the suppressed than for the baseline items. And that was also the case in the right parahippocampal cortex. So suppression indeed seems to lead to a sustained reduction in cortical reactivation. Now, we had further hypothesized that the suppression-induced reduction in scene reactivation constitutes the neural basis of the reduction in vividness, right? And we therefore expect a positive association between these measures. Now, to test this account, we calculated for each participant a score of the suppression-induced reduction in vividness. So this score was calculated as the reduction in vividness from the pre to the post test for suppressed memories, corrected for the reduction in vividness for baseline memories. And we did this for the reactivation, and we did calculated the scene, the same score also for the vividness, right? And this is a scatter plot showing you how the reduction in vividness relates to the reduction in scene evidence. And I'm here using then robust Spearman skip correlations, which would essentially disregard the contribution of outliers. So in the left hemisphere, we see a trend for a positive association between these measures. And this effect is indeed significant in the right parahippocampal cortex. So a greater reduction in scene evidence is accompanied by a greater reduction in the vividness with which the memories are being experienced. Right. Yeah, so then essentially let me conclude that I hope they've shown you some evidence that memory suppression can reduce the vividness of aversive memories, and at the same time reduce global and local reactivation of pertinent information. And there's an association between these two phenomena, which suggests that sort of, this sustained reduction in reactivation constitutes the neural basis of suppression-induced forgetting. And I'm not going to show you the data today, but essentially we find a similar association between these effects when we are not just looking at global reactivation, but use RSA essentially to look at the reinstatement of unique memory representations, even though we did not find the main effect. But yeah, I could talk about this. So yeah, so there's yeah, this is the preprint which we are about to update, and this as well as the other paper also has links to the data and analysis scripts on OSF, of course. All right, with this, I would like to conclude overall that memory suppression, I hope to have shown you, can be achieved via two mechanisms that get transiently engaged to prevent retrieval, and they then have sustained after effects. So they indeed cause forgetting in healthy individuals. And now we know that it helps to tell people what to do. Also in this case, this seems to be deficient in some disorders characterized by intrusive thoughts. This may reflect also an inability essentially to reactivate the neural responses. And I think that together these data nicely corroborate Mike's groundbreaking observation that suppression causes forgetting, and further highlight the clinical relevance of this process. Yeah, let me then just conclude by highlighting my awesome team here in Leipzig, in particular also Ann-Christine Meyer and Davides Samaja, who did the work, and of course my collaborators, in particular Mike. And again, my congratulations to this award, given that this is only mid-career. Right, I really can't wait to see what's yet to come in the second half. So congratulations again, and thanks everyone for your attention.
Thank you so much, Roland. You stole my line about the mid-career prize. So before I read any question, if you have any question, please ask to run it in the Q&A box. But in the meantime, Roland, I have one question for you. Did you look at whether the initial vividness is linked to the neural marker of suppression, either in terms of coupling or local suppression activity in the hippocampus? Because we might expect that greater vividness initially might require more inhibition, more control?
Yeah, I mean, that's a good question. We also played around this with the idea that with the question, what degree of reactivation? So there's the idea, right, that how can transient suppression actually lead to forgetting, right? If you don't think about the memory, how can the memory essentially get compromised, right? And then obviously there are some ideas reflected in some of the work on intrusions that Mike has done, but also by Kahneman's work, that actually for memory to be liable to disruption, it needs to receive a certain level of reactivation, right? Yeah, so we wondered if we could use this for to essentially see how strongly a memory is reactivated, to see whether there's a subsequent effect on this. But the study wasn't really set up for this. What we do know is we did look into our classifier quite a bit, right? So for example, we ensured that on the pre-test, right, when people have not messed around with the memories, we can show that a stronger scene evidence correlates with a stronger subjective vividness, right? So yeah.
Okay. Thank you so much, Roland. I don't see any question. Oh yes, one question. So from Tara Zach Saite, apologize for the mispronunciation, Tara. Very interesting talk and findings. I wonder whether you would be able to share any thoughts related to the direction of effect related to anxiety. Do you think it's a memory suppression or lack thereof effect may maintain anxiety, or would higher anxiety lead to less suppression? So the chicken and egg question, Roland.
Yeah, I mean, we can answer with it. It's hard to answer with this with these data, and I think, well, Pierre, you may have a better answer later today. I mean, really, to do this, we would need a prospective study, right? You know, to see if if some sort of baseline ability of suppression would allow you to be protected against the aversive consequences of traumatic experiences. However, what I think there is some evidence, though, that memory suppression is also something that people can train to in some respect, right? So there's a really nice study by Justin Halbert, a former graduate student of Mike, who have, Mike, tell me if I'm wrong, but my recollection is that they essentially have a large sample of undergraduate students, and essentially, in addition to their ability to suppress memories, they also assess the number of aversive life events that they had experienced in their past, right? Yeah. And what they observe is that those people who had a stronger trauma load, I think it wasn't strictly traumas, right? But that they essentially show a better capacity to forgetting, right? So now these would presumably be kids that actually have had more anxious events, right? And more anxiety experience in their life, and yet they show better memory, right? Suggesting that it may not be anxiety per se that reduces the ability to suppress, but that it is maybe actually the other way around, that if you have more events in which you have to deal with, that you can actually train sort of this capacity to some effect. Yeah.
Okay, thank you so much, Ron. Yes, thanks a lot. I think we are now going to move to the next talk. That will be, it's my great pleasure to introduce Dace Abzvalka. So sorry, I have trouble with name pronunciation today. Sorry, Dace, if I mispronounce your name. Dace has started a career in computer system, and then she decided to move to neuroscience. And after a PhD from Bangor University in Wales, she moved to the CBU to work with Mike as a postdoc. And after a postdoc, that ended this year, this summer. She stayed at the CBU and now works in the method group at the CBU. So welcome, Detje.
Hello, everyone. Can you see my slides?
Yes, we can.
Okay, thank you. So yeah, congratulations, Mike, on your achievements that bring us all together here today. And thank you, Pierre, for organizing this celebratory symposium. I will present a project which I did as a postdoc in Mike's lab in the last couple of years, and which should come out published next week. Actually, I just received an email from publishers that we are scheduled to be published on 12th of January. So please check my Twitter for announcement. And this work extends the memory control to a more broader question of domain general inhibitory control. And we look at the parallels of inhibiting unwanted memories and unwanted actions. The ability to stop unwelcome thoughts and memories is essential for our well-being, as we already heard in the previous talks. And Mike's work has focused on mechanisms of memory control and suggests that this vital ability may be grounded in inhibitory control. Inhibitory control is also behind our ability to stop unwanted physical actions. We stop our hand from touching a hot stove, and we also stop intrusive thoughts to avoid pain. And both stopping actions and stopping memories are a form of inhibitory control, a supposed domain general control mechanism supported by the right lateral prefrontal cortex. The concept of inhibitory control has attracted much interest in psychology and neuroscience over the last 2 decades. But if there is a domain general inhibitory control, how does it flexibly inhibit diverse content processed in diverse brain areas? You see, memory retrieval and consequently retrieval stopping involves the hippocampus. And Mike's work has shown that the right prefrontal cortex causally suppresses the hippocampus to prevent the retrieval of unwanted memories, as Roland also mentioned in the previous talk. But on the other hand, work on motor control shows that motor actions involve the primary motor cortex, and prefrontal cortex suppresses primary motor cortex M1 to prevent action execution. So stopping actions and memories would require that the domain general inhibitory control region targets domain-specific brain areas to suppress motoric or mnemonic processing. And no study has shown such dynamic targeting by domain general control region. And here in this study, we tested the existence of dynamic targeting by asking 24 participants to stop unwanted actions or unwanted memories and scanning their brains while they were doing so to see what's happening. For memory inhibition, we used the think-no-think task developed by Mike almost exactly 20 years ago. And for action inhibition, we used another classic task, stop signal task, developed by Gordon Logan in 1980s. So in the stop signal task, participants learned to associate different colored circles with the left or right button presses. And during fMRI acquisition, participants perform speeded motor responses to the presented color circles. And these responses are really automatic for them. They see the color and they press the button. But occasionally, shortly after they go stimulus, a stop signal occurred, and participants had to withhold the initiated response. So they were ready to press, but they heard the stimulus and had to withhold the response. The ability to stop the motor response was measured by the stop signal reaction time. And the shorter the stop signal reaction time, the better the motor stopping ability. And in similar parallels, in the think-no-think task, participants form associations between 60 different word pairs, such as part ball, spray cord, radio tree, which are then grouped into think, no-think, and baseline conditions, 20 word pairs per condition. Then during the fMRI acquisition, participants see a keyword only. And if the keyword is presented in green, they are asked to retrieve the associate word. And if the keyword is presented in in red, participants are asked to inhibit thinking of its associate word, and if it pops into their mind, to actively push it out and not think of it. Finally, there is a cued recall memory test where participants attempt to recall all initially learned associations, and typically recall performance suffers for no-think items compared to baseline items that were neither retrieved nor suppressed during no-think phase. And this difference between the correctly recalled baseline and no-think items is known as suppression-induced forgetting. It indirectly measures the ability to stop unwanted memories. And the larger the suppression-induced forgetting, it indicates better memory suppression ability. And we were particularly interested in the inhibition mechanisms for these two tasks. Throughout the fMRI acquisition, our 24 participants performed short 30-second mini blocks of stop signal and think nothing tasks, thus dynamically switching between the action and memory modalities. In each fMRI run, there were 8 such blocks, and altogether there were 8 fMRI runs. Our aim was to test the hypothesis of dynamic targeting as a mechanism that enables domain general inhibitory control of actions and memories. Dynamic targeting requires that a candidate inhibitory control system exhibits certain attributes. We defined and tested 5 such attributes. First, there should be, of course, a domain general inhibitory source that engages the proposed source of control. An activity in the inhibitory source region should generalize over the specific demands of each stopping type. And we predicted that such inhibitory control source would be in the right lateral prefrontal cortex. To identify the domain general inhibitory source, we first performed a whole brain univariate conjunction analysis, looking at the conjunction of stop greater than go and nothing greater than think contrasts. The conjoint activations of both motor and memory inhibition were mostly right lateralized, with conjoint activation clusters in both dorsal and ventral lateral prefrontal regions. I will now focus on the right hemisphere only. Remember that our design involved rapid alterations between the mini blocks of the stop signal and think nothing task. So it could be that we see these conjoint activations due to some carryover effects between tasks due to the rapid dynamic switching. To examine this possibility, we compared the activations observed in our within subject conjunction analysis to a meta-analytic conjunction analysis of independent between subject stop signal and think nothing studies. This meta-analytic conjunction results were very similar to our within subject results. Here's the overlap shown in yellow, and the overlapping domain general activations were both in the right ventral lateral prefrontal cortex, Brodmann areas 44 and 45, and in the anterior portion of the right dorsal lateral prefrontal cortex at the junction of Brodmann areas 9, 10, and 46. The convergence of this conjunct analysis suggests that our findings of conjoint activations across the two inhibition modalities do not just arise from some carryover effects due to our dynamic within subject design. And overall, the findings of this univariate and meta-analytic conjunctions confirm the right lateralization of inhibitory control and the role of the right ventral lateral prefrontal cortex and dorsal lateral prefrontal cortex in motor and memory inhibition. Nevertheless, it is possible that despite the shared engagement of these regions, the patterns of activation across voxels is different for the two modalities, which would then undermine the domain generality claim. Thus, to test the domain general inhibitory mechanism even further, we assessed whether the two inhibition modalities evoke similar activity patterns within the dorsal and ventral lateral prefrontal regions. So is the stopping actions pattern activity, voxel pattern activity, same as stopping memory, voxel activation pattern activity? So within each of these two prefrontal regions, we trained a classifier to distinguish inhibit from respond voxel activation patterns in one modality, for example, distinguish no think from think, and then tested this classifier whether this trained classifier could distinguish inhibit from respond in other modality, that is stop from go. And in both dorsal and ventral regions, the classifier could cross-classify the inhibition conditions reliably above the 50% chance level. This suggests that the two stopping tasks not only increase overall activity in the DLPFC and VLPFC, but also generate common activation, voxel activation patterns. So interestingly, if we look at the cross-classification across the 8 fMRI runs, we see that the classification accuracy is higher when participants start the task and gradually decreases over time. This linear decrease was observed both in the right DLPFC and to a lesser extent also in the right VLPFC. That means that the two stopping processes, stopping actions and stopping memories, are more similar initially, but become less similar over time. We had in fact predicted this decline, that's why we looked at it, and it is consistent with adaptive forgetting. And as participants try to suppress the memory fades away, and less suppression, less inhibitory control is required. This successful memory suppression requires less inhibition with time and becomes less similar to action inhibition. So the univariate meta-analytic and multivariate analysis confirmed the first attribute of the domain of the dynamic targeting hypothesis. Specifically, we identified two domain general inhibitory source regions in the right prefrontal cortex, the dorsal and ventral regions. These regions not only increased overall activity when stopping actions and memories, but also shared common voxel activation patterns for the two stopping tasks. The second attribute of domain general dynamic targeting requires that the engagement of the prefrontal sources demonstrates their behavioral relevance. That is, the activity in the domain general inhibitory source regions should be related to the ability to stop both actions, as measured by the stop signal reaction time, and memories, measured by the suppression-induced forgetting. First, we wanted to see the relationship between the two behavioral measures. Is the action stopping efficiency related to the ability to inhibit unwanted memories? And we looked at the correlation between the stop signal reaction time and suppression-induced forgetting. Here, the lower the stop signal reaction time indicates better action inhibition ability, and higher suppression-induced forgetting indicates better memory inhibition ability. And our data showed significant correlation between the two inhibition measures, such that better action inhibition was related to better memory inhibition. These behavioral results suggest that there is indeed a common inhibitory process for stopping actions and memories. And people who were good at controlling their memory, their motor actions, were also good at controlling their thoughts. Next, we wanted to see how the inhibitory control activity relates to the behavioral measures of inhibition. We hypothesized that if the areas showing conjoint activations of motor and memory stopping supported the domain general inhibitory process, activation in the very same voxels would predict both action stopping speed and suppression-induced forgetting. To test this, we performed partial least square analysis. We looked within the meta-analytic stop and nothing conjunction regions and identified voxels in our within subjects data where a higher bold signal during inhibitory control predicted both action stopping speed and better memory inhibition. So the red areas here had the this behavioral relationship with these activations, better action inhibition and better memory inhibition. And such brain behavioral relationship was found in both DLPFC and VLPFC voxels. That is, increased activity in the very same voxels within the DLPFC and VLPFC predicted both better stop signal reaction time and better suppression-induced forgetting. Another proof of the behavioral relevance of the two domain general source regions, we already saw a few slides earlier in the multivoxel pattern analysis. There we observed that the two stopping tasks became less similar to each other with time. We explained it as successful adaptive forgetting, when successful inhibitory control of a memory requires less control with time. This relationship was confirmed also with the behavioral data, that is better memory inhibition leading to forgetting and measured by suppression-induced forgetting was correlated with steeper decline in the voxel cross-classification accuracy. And this relationship was strong for both source regions, and these are showing robust correlations. The classification accuracy decline was also correlated with better action stopping ability. Thus, the stopping representations in the right dorsolateral prefrontal cortex and right ventrolateral prefrontal cortex were related to both the ability to stop memories and actions. Thus, we confirm the behavioral relevance of the domain general inhibitory source. The next set of attributes of domain general dynamic targeting requires domain-specific targets, which we a priori chose to be M1 for stopping actions and hippocampus for stopping memories. The third dynamic targeting attribute we tested was the suppression of function in the target regions. We wanted to verify that stopping actions and stopping memories downregulate modality-specific target areas. We hypothesized that inhibiting memory retrieval would downregulate activity in the hippocampus, but inhibiting motor responses would downregulate activity in the primary motor cortex. We looked at the both signal difference between the inhibit and respond conditions in each modality, and we found a significant interaction between the inhibition modality and the modulatory target regions. Specifically, stopping motor responses evoked greater downregulation of the M1 than the hippocampus, and stopping memory retrieval evoked greater downregulation of the hippocampus than M1. And critically, these modulations were not just due to upregulation in the go or sync conditions, but as illustrated here in this slide, by negative bold responses during stop and no-sync conditions, the domain-specific target regions showed evidence of interrupted function during stopping, which is consistent with the requirements of dynamic targeting. So thus we confirmed our third attribute that there is a distinct activity suppression for stopping actions and memories. The fourth attribute of the dynamic targeting hypothesis is that there should be a causal modulation by the source. That is, the domain general source should exhibit a causal influence over the target region, suppressing its function. And furthermore, the fifth attribute is that this causal modulation should be goal-dependent. That is, domain general inhibitory source causally influences M1 when inhibiting actions and the hippocampus when inhibiting memories. To test this fourth and fifth attribute, we tested the causal relationship between the two domain general inhibitory source regions and the two domain-specific target regions. And we tested 73 possible models of how these four regions might be connected to each other. Specifically, we used dynamic causal modeling to compare the model evidences for 73 models. Here, I just quickly flash an overview of our model space of possible interactions that we tested. And from the 73 possible models, our data showed one quite strong winning model with 0.92 probabilities that this model is more likely than any other model tested, given our data. And specifically, what this model indicated was that during action stopping, the DLPFC and VLPFC interact with each other, and both of them modulate M1 activity. But during memory stopping, also DLPFC and VLPFC interact with each other, and both of them modulate hippocampal activity. This result suggests that both right dorsolateral prefrontal cortex and ventrolateral prefrontal cortex are similarly involved in the domain general inhibitory control, and they both dynamically target the domain-specific regions to inhibit their functions based on their supporting goal. Thus, we have confirmed all 5 attributes required that we defined that are required by the dynamic targeting hypothesis to be true. We identified domain general inhibitory source in the right lateral prefrontal cortex, the right dorsal and ventral prefrontal cortex, and both seemingly equally important. We confirmed that activity in both source regions was related to the behavioral performance of both action and memory stopping. And activity in the domain-specific target regions, M1 and the hippocampus, was suppressed in line with the stopping goal. And the domain general source region causally modulated the target region, and this modulation was goal-dependent. The source The source is dynamically targeted M1 for stopping actions and the hippocampus for stopping memories. The main conclusion, and also the title of this work coming out next week, is that dynamic targeting enables domain general inhibitory control over action and thought by the prefrontal cortex. So what are the implications of our findings? Well, dynamic inhibitory targeting can account for both association and dissociation in the abilities to suppress unwanted actions and thoughts. On the one hand, deficits in both action and thought stopping should arise with dysfunction in the DLPFC and VLPFC in the control regions, given the domain generality of these regions. For example, people scoring highly on impulsivity also report greater difficulty with intrusive thoughts. And obsessive thoughts and compulsive actions are present in obsessive compulsive disorder, and intrusive memories and impaired response inhibition is present in post-traumatic stress disorder. Persistent worry and impulsivity in anxiety disorders, intrusive thoughts and compulsivity and addiction. And these comorbid deficits may reflect dysfunction in the dorsal prefrontal cortex, the ventral prefrontal cortex, or in the other shared components of their control pathways. On the other hand, dissociation in the abilities to suppress unwanted actions and thoughts should arise when dysfunction selectively disrupts the domain-specific target regions, M1 separately or hippocampus. For example, individual variation in local GABAergic inhibition with the hippocampus or M1 within the hippocampus or M1 predict inhibitory control over memories and actions independently of prefrontal function. These separate contributions of domain general and domain specific factors to inhibitory control implied by dynamic targeting limits the utility of, for example, using motor inhibition as a metric of inhibitory control over thoughts. This might explain some findings in the literature that show surprisingly small stop signal reaction time deficits in major depression and anxiety, where the deficit might rather be in the hippocampal, not the prefrontal function. On the other hand, greater stop signal reaction time deficit is observed in attention deficit hyperactivity disorder or obsessive compulsive disorder, where this dysfunction might rather be in the prefrontal control, not the individual target regions. So as I mentioned, the present study will be published next week on Wednesday, as the publishers say. Please check my Twitter for an announcement. And this work was done in collaboration with Katerina Ferreira, Taylor Schmidt, James Rowan, of course, Michael Anderson. And congratulations again, Mike, and thank you for the opportunity to do this work with you and to present this study to you all. Thank you.
Thank you so much, Lecce, for presenting to us this fascinating data. There is 2 questions in the chat for you from Elizabeth Runis. Thank you for your very interesting work. Have you analyzed the patterns in BA40, which also seems to be part of this network? This is the first question. And the first question from Elizabeth is, what happens in the left hemisphere? Question mark.
Okay, so I will first look at this. So have we analyzed? Yes, so in the parietal cortex, we in fact indeed looked also in the others, these domain general regions that we identified. And yes, there was the same domain general cross classification also in area 40 in both in the parietal cortex, yes. But there wasn't, if you look at differences, there wasn't this what we tested later.
Was he also linked to behavior?
There wasn't the behavioral relevance to the.
Okay.
So what was the other question?
Yeah, the other question was.
What happens in the left hemisphere? So
yes.
In the left hemisphere, there was only the left BA40, so the left parietal cortex, which showed the same results as the right parietal cortex. That there was a cross classification significant, but not the POS results, not the behavioral relevance. So we just looked at it's included, I think, maybe in our supplementary results of the study. Yeah. Also. Yeah.
Thank you, Deja.
But only these two prefrontal regions showed like the cross classification and also the behavioral relevance of the parietal regions.
Oh, and another question for you from Gonzalo Ursula. Hi, Gonzalo. Excellent work and very well presented. That I wonder why, if there is a single domain general mechanisms, two prefrontal subregions are involved.
Yeah, that's a good question. It's like in these things that we tested, this both seem to be equally involved, but of course there might be some differences that we didn't test, like attention-related differences, or, well, I don't know. I'm afraid I can't answer this question. I think there might be some commonalities that we found in the two regions, but there probably are also some more specific functions that the two regions do distinctively, but that's still to be found out. I think Mike probably knows better all this literature around these two regions, and there might be some indications. I know that there are some talks about attention involvement, or so. Yeah, it's hard to say, but I suppose there are commonalities, and we only found commonalities, but we only looked. We were trying to find commonalities. We didn't even try to find differences. So there must be also something, some sub-functions that the two regions do separately.
Okay. Well, with that, I think we can move on to the next talk, which is myself.
So I'm just looking for the stop sharing.
Yeah, stop sharing the screen, unless there is more question in the chat.
Stop share. Okay.
Okay. So.
Can I just mention, Pierre?
Yeah.
Can you hear me?
Yeah.
Regarding Gonzalo's question, hi Gonzalo, by the way. The two regions that Dots identified are both part of the singulo-opercular network. So it's they might look like just two disparate, unrelated regions, but in fact, in large-scale brain network analyses, they're not unrelated. So it's a question of how all that works together is something for future work.
Thank you, Mike. Okay, well, thank you very much. And while you are here, I take the opportunity again to warmly congratulate you for this very well-deserved prize. As Roland, I'm looking forward to the second part of your career, because this is a mid-career prize. I'm really much looking forward to that. I would like also to thank you because I was, I should say that I was postdoc with Mike, and really I learned the ropes with with him, and really had fantastic time working with him. Learn a lot of skills and competence, also together with my other mentors at the CBU, Matt Davis and Rickenson. And the training I've received there with you guys was absolutely fantastic. And really enjoy so much the world of forgetting that I decided to export it to France, basically. And today in today's talk, I'm going to ask a question. I'm going to try to explain you why this question is surprising, and I'm going to try to convince you that this question is actually important, and show that this question is directly issued from Mike's works and Mike's idea on suppression and forgetting. And I try to show that actually his idea on active forgetting actually completely changed the way you should understand PTSD. And I'm going to try to explain why. So a lot of things have been already said, so I'm gonna really move very quickly on the introduction. As you know, memory is super important to adapt, adapt your behavior. It's important to recall positive memory, to note repeat and adapt your behavior, but also negative memories, all this, obviously, you know all that. Sometimes, however, memory becomes non-adaptive. That means that you recall a memory in a context that is not relevant. And this non-adaptive memory question us on the role of adaptive forgetting. So in the past, forgetting has largely been viewed as a side effect of memory. So you forget because of memory. So you forget because you do not reactivate your memory, or you forget because new memories create interference and forgetting. So forgetting was just somehow the side effect of memory. And I think
you haven't started your presentation yet, Pierre.
Sorry?
You haven't started your presentation yet?
I did.
Something, Roland also just said something in the chat.
Oh, sorry. So you can't see my slide?
We can see it in presenter mode.
Yeah,
so you shared the wrong, see if you shared the wrong screen.
Okay, I'm going to start again. Sorry for this guy. Thank you for letting me know. Is this better now?
Yes.
Okay. So this is what I was saying about adaptative and non-adaptative memory, and about the role of adaptative forgetting. I guess you get the idea. I don't have to repeat myself. And I was saying that, you know, before Mike's works, forgetting was largely viewed as the side effect of memory because of memory decay and interference. And the big contribution, obviously, of Mike's research was to show that forgetting can be an active process, an active mechanisms depending on inhibitory control. And I highly recommend to read the recent reviews they wrote with Justin Albert that really highlight this question very well. So I'm not again going to present the think-nothing paradigm. I think you now understand how this paradigm works. You know, when after learning, you have to suppress on your conscious awareness memories by focusing on the paying attention to the reminder cue. And today I'm only going to talk about direct suppression instruction. So when you have to really focus on the cue, blank your mind, and do anything you can to push out of awareness the memory if it pops into your mind. However, going to introduce a slight modification compared to what we've seen before in the paradigm, with the notion of intrusive report, subjective intrusion report. This was introduced by Ben Levy and Mike in the 2012 study. And basically, the idea is that you can ask the participant after the end of the trial whether he had, even if it's extremely brief, an experience of an unwanted or intrusive memories that pop into mind despite his effort to try to push it out. And making a long story very short, there's many data that suggests that this intrusive memory creates extra inhibitory demands and provoke more forgetting and more control in general. So my works in the past has largely been focusing on what's going on when we try to suppress this intrusive memory. And what we find with Mike is that you actually have, as I was saying, an increase in inhibition that is reflected by a greater negative top-down coupling between the control system in the right MFG and the target region. So the target region here can be the hippocampus, obviously, to stop pattern completion and memory retrieval, but also, regions related to specific features of your training material. So, for instance, at the amygdala, if this is aversive scene, or the visual system, if this is objects, for instance. And what you see here in the bar on the right is the greater inhibition reflected by more negative coupling, suggesting that the MLG is somehow stopping or interrupting the activity in this target region. So, okay, so what are PTSD? So obviously, intrusive memory is there is the central features of PTSD. So they have extremely vivid, intense images of the trauma. There is usually some kind of hotspot of some specific moment of the trauma that are extremely stressful for them, provoke a lot of anxiety, a lot of fear. And as a result, they always scanning the environment, have some hypervigilance behavior, and ultimately will have some form of avoidance to try to avoid the reminder of the trauma. I'm not going to go into the detail of this, but also this intrusive and the stress induced by this intrusive memory can cause a lot of damage in the brain due to the stress and provoke, for instance, some form of hippocampal atrophy. So there's many neurobiological models of PTSD in many ways, but somehow they all kind of agree on the fact that there is two main routes to develop a traumatic memory. So why do you develop a traumatic memory? Why do you develop intrusive memory? Well, the popular model proposed by Chris Burin is that there is two routes. One is a form of what we call over-consolidation and hyper-consolidation from the amygdala to the dorsal stream that will somehow crystallize some specific sensory images and emotion linked to the original trauma and provoke these extremely stressful images. And this will also be linked with a poor contextual integrations related to the dysfunctioning of the hippocampus. And what this poor contextual integration does is mean that any cue, even weakly related to the trauma, can trigger this intrusive memory. And also the poor contextual integration gives you the sensation that it's happening again in the present, because you can't put this memory in the past, because the poor contextual integration gives you the sensation that you are reliving the scene again in the present, which is extremely scary for the people. And my work, that originate from Mike's idea, is that actually, and I should mention, sorry, that obviously this lack of contextual integration fits well with the observation that there is a hippocampal atrophy in PTSD, which is a really strong observation. And my works, inspired by Mike's works, is to propose that perhaps inhibitory control deficit, which has nothing to do with memory disorder, also contribute to the formation of this traumatic engram. And I tried to do some digging to see where this idea comes from in Mike's original paper. And the first mention of this idea I found was in this paper that you wrote, Mike, with Ben Levy in 2008. Maybe there is an earlier mention of this connection. And this idea, I think, originated from the observation that there is some individual variability in the capacity to control memory, and that perhaps this strong individual intervariability can be related to why people, the variation in response to trauma, and why some people develop PTSD and others are not in response to the trauma. So to test this idea, we launched in 2015 the project that we call Remember, which was launched after the terrorist attacks that took place in Paris in November 2015. It's a project that is a longitudinal project that we start data collection in 2016, so a bit around a year after the attack. We've seen them 3 years after, and we're going to see the same participants again very shortly. This is a multimodal imaging project. We collect many types of data. Obviously, the TNT task I'm going to present, but also hippocampal subfield, high resolution images to delineate hippocampal subfields, DTI data to study the connectome, and resting state data, and so on. And this project is articulated around a larger project that we call the November 13 programs. And at the heart of this program, there is a study that was the Study 1000, in which they collect and record interview of people more or less connected to the attack that recall and explain what happened to them, but also the consequences on their life and so on. And this is all stored in National Archive. So it's a transdisciplinary project that has many ramifications in terms of big data, history, sociology, collective memory, and so on and so on. But this, you have to know that this Remember program is actually collecting data on the same participants that participate in this larger program. And we've seen 120 exposed participants to the attack and 80 non-exposed participants. At T1, we had about 50% of the people who were what we call resilient, 50% of the exposed that did not develop any sign of PTSD. And 25%, 50% that were what we call PTSD plus. So among these 50%, half of them were what we call partial PTSD, meaning that they have intrusion, strong impact on their functional life, on their social life and functioning. They can't go to work and so on. And they also have other symptoms, but they don't have the full list of PTSD symptoms, but they have really the intrusion. And it's not a very well recognized and validated clinical concept, this notion of partial PTSD. And the other half are full PTSD with the full symptoms. So, as you can imagine, we wanted to know the role of memory suppression in this variation in response to trauma. And this work was performed with Alison Marie, was my postdoc at the time. And in this study, we use completely neutral, we use pairs of words and objects with completely neutral pictures, and we focus our study on the connectivity between the control system and between the memory region, using both functional and effective connectivity analysis, contrasting what's happened during intrusion compared during non-intrusion attached with a no-sync reminder queue. What we observed was quite clear-cut that both non-exposed and exposed individuals without PTSD were perfectly capable of down-regulating memory and hippocampal activity using the control system. This was clear both in the functional PPI analysis, but also in the DCM analysis. And this strong down-regulation for intrusive memory compared to non-intrusive memory was absolutely not present for PTSD. Suggesting that this alteration in this compromission of the control system may have contributed to the development of their disease. However, what this study really doesn't show is the origin of this control disruption. And for the rest of the talk, I'm going to try to bring a new idea that this disruption can be linked to what I've called predictive control. So what's going on when you try to, the initial idea was that if you have an intrusive memory that is a form of memory signal that tells the control system that you need to downregulate memory activity, this is what you observe here with standard marker of connectivity. You observe more suppression, more downregulation from the control system to the hippocampus for intrusion compared to non-intrusion. Okay, this is a standard scenario that we envision when we do this study. But actually, as you know, the brain is a statistical predictive machine. So the brain always tries to predict what's going on and what will happen next. And this is especially true in the sync-no-sync paradigm because you have a series of memory cues, and sometimes they're attached to intrusive memory, sometimes they're not attached to intrusive memory. And it's very likely that the memory system tries to anticipate and predict for each cue whether or not it will have some kind of intrusive memory. So the participants have to form some kind of belief of whether or not you will experience this intrusive memory based on the past occurrence of the trials. Okay, so the problem is that, and obviously, once you form this belief, if you have an intrusion, that causes an error in the system because this is not necessarily matching what you have anticipated. So you have a belief signal that you will have an intrusion, but you also have a prediction error signal that say whether or not your belief were correct. And the problem is that if you, so in the case of the normal inhibitory control functioning system, you can see that both signal might contribute to the observed pattern where we don't dissociate this hidden computation, if you like. But in the case of dysfunction of disrupted control, actually, an alteration of the predictive control signal or of the predictive control mechanisms or an alteration of the reactive control mechanisms and different combination of alteration of these mechanisms can cause the pattern that we initially observed in PTSD. So we wanted to we wanted to actually explore this idea and test whether the disruption, the origin of disruption we've seen was linked to whether one of these components in PTSD. But the problem is that we don't know this belief, right? So we don't observe them. So we only have the behavior of the participants that tell us whether he had an intrusion or not. So we had to develop some kind of mathematical modeling to infer what are the beliefs of the participants about the fact that he's going to have some intrusive memory based on this behavior and based on the history of the trials. And this is what we do with Giovanni Leone, who was a PhD, who just finished his PhD and now is moving to postdoc with me. And Giovanni developed this mathematical modeling of intrusive belief. And I'm not going to go too much into, I'm not going to go into the detail of this model, but basically, I'm just going to show you here, for instance, the output of this model. So here, this is the dots are intrusive trials when this is 1, and non-intrusive trials when this is 0 here. So this is a series of participants reporting whether they had intrusion or not. And the blue line is the beliefs that the model actually learns from this pattern of behavior. So the model basically updates his belief at each trial based on the history of the previous trials. And the prediction error caused by the new trials update actually the beliefs of the participants, but update according to the amount that depends on the uncertainty that is calibrated in this model. So basically, this is some kind of meta-observer model that learn all the participants from belief about the way he behaved in the task. And so the blue line is the belief itself about intrusion expectation, if you like, and the red line is the error caused by the presence or the absence of an intrusion. What we did with Giovanni, we then use these computational indexes and put them into DCM to do what we call computational dynamic causal modeling, if you like. And we input this computational index over the coupling that modulate activity from the MFG to the hippocampus to see all this belief and prediction error about intrusion modulate the coupling between the control system and the hippocampus and also the precuneus. We were quite, I think, to be honest, I was quite astonished by the result, and I find the result quite fascinating. So what you see here in red is a prediction error, the control of prediction error. So negative value means that you suppress according to prediction error, and for the red, and for the green-blue, negative value means that you suppress according to belief. So the more it's negative, the more you suppress your belief for the green. You can see that there is a perfect balance between the two in non-exposed and PTSD. So they are capable of balancing control of prediction error and control of belief. And this imbalance is absent in this balance is absent in PTSD, and we see an imbalance. So they basically only control belief. So they control something that does not exist. They control not the error and the intrusive online reactivation of intrusive memory. They actually control the belief that something will happen. And very interestingly, this imbalance that we see in PTSD is specifically linked to the cardinal features of PTSD. So it's mostly linked to intrusion and avoidance, but it is not linked to other factors like anxiety or affect. So just to try to illustrate a bit more this idea, I'm going to use one of my heroes, Federer. So imagine, Federer, when he wants to control and send back the ball to his opponent, he has to somehow predict where the ball will land in order to adjust his step toward the ball. If you use too much his belief, then he will likely be wrong and will have to adjust too much. And if he doesn't use his belief at all, then he will have to react too quickly to the ball. So a very important aspect for him is to balance well his belief about where the ball will land and his capacity to react to the ball in case he has a slight error in his belief. So I think if you replace now the ball by an intrusion, I think it's exactly the same thing. So you need to have a perfect balance, a good balance between controlling the intrusion based on your belief that they will happen, but also based on the fact that when they're actually reactivated, you need to be able to adjust and react to this error. than they cause. Time. So now to conclude, and I just would like to try to maybe dwell upon or elaborate a bit more on this idea that inhibitory dysfunction can cause the persistence of the trauma, because what I've showed you is just from a cross-sectional study. There is absolutely no evidence that what we observe can cause the development of the traumatic memory. So I can only speculate on this, and I'm going to try to speculate on why I believe inhibitory control dysfunction that may be pre-existing, but also that probably not only pre-existing, but also induced by the stress on the system. So very, very likely this inhibitory control dysfunction, even though they might slightly pre-exist, they are very likely exacerbated by the effect of stress. So I will try to elaborate on why such dysfunction can promote, if you like, the formation and the maintenance of the traumatic engrams. So the first idea is linked to predictive control. Why predictive control is not that good? Well, obviously, it preserves the memory trace. When you control a belief, you don't actually control the memory trace, and you just leave the memory trace intact and not the memory itself. Another interesting hypothesis, just a speculation, is that if you have applied excessive interruption of hippocampal processing, you might also induce some form of contextual amnesia. And this contextual amnesia is also a landmark of PTSD. So PTSD, they don't have memory for the context, they only have memory for a very specific hotspot in the trauma without any context. So if you suppress too much based on predictive control, maybe you prevent the participant, the person, to learn the new context, if you like, and to update his memory with new context. And with reactive control, I think it's very interesting, this link between prediction error and the role of prediction error, because there is more and more evidence to suggest that prediction error can increase memory destabilization. And the control of prediction error might promote forgetting according to two distinct hypotheses, which I would like to quickly suggest to you today. The first one is linked to the disruption of raw consolidation, if you like, and the second one is linked to increasing what we call the inhibitory engram. So as Maria proposed and suggested at the end of her talk, she mentioned the fact that retrieval is a form of consolidation, right? So when you retrieve memory, you recreate a moment where you can reconciliate and strengthen the connection in the neocortex. And we know actually that this reconciliation is linked also to prediction error. It's promoted by the presence of prediction error. So in these frameworks, it's easy to understand that if you suppress hippocampus, then you stop this reconciliation process that happens during the recall of intrusive memory, and you kind of degrade and weaken your memory. If you don't have this inhibition capacity, then it's easy to envision the fact that you will again promote the reconciliation of your memory, because you don't have these skills and the inhibition, power, if you like, to stop this reconciliation from happening. And by this view, that means that what I like about this idea, that this means that suppression will directly inhibit the memory engram itself. So that's the first idea. When you suppress the memory engram. That's one hypothesis, right? But there is also an alternative hypothesis, which is actually that you don't suppress the memory engram, but you just silence the memory engrams. And this idea comes from the work of Helen Baron on the inhibitory engrams, which I think proposed an extremely interesting framework in which she said that you recall a memory by disinhibiting this inhibitory engram. So when you have excitatory engrams, and for each excitatory engram, you have a mirror of inhibitory engrams. And the recall is actually disinhibition of this inhibitory engram. So you don't recall by activating your memory, you recall by disinhibiting this inhibitory engrams. Which means that when you forget, you just that the memory is not disappear, is simply silenced. Is silenced by this inhibitory engram. So again, this framework, if you can suppress the hippocampus, because this is the hippocampus that doing the disinhibition, then you just keep your memory silent. the memory is not suppressed, it's just silent. And again, if you can't inhibit, then you reinforce and actually prevent this inhibitory, prevent the maintenance of this inhibitory plasticity. But these two ideas are really interesting because one proposes that you actually suppress the engram itself, and the other proposes that you actually just put the memory into silence. And it also has a lot of interesting consequences. So just to conclude, and some take-home message of how communities use that inhibitory control mechanisms linked with active forgetting are central to understand the variation in response to trauma. The question is whether this is something pre-existing or with something post-traumatic. The imbalance between reactive and predictive control seems to play a fundamental role to understand PTSD, but we need to clarify the relationship between these two mechanisms and the lack of forgetting and the trauma persistence. The treatment focus on inhibitory control could complement existing one using material completely removed from the trauma, but then it raises the question of how do you train a compromised system, right? And finally, one very important question is all these findings are inculcated in the observation in PTSD that there is hippocampal atrophy, hippocampal dysfunction, and this is also a very interesting idea that Mike has suggested with Taylor Smith to investigate the role of the GABA system, and especially GABA in the hippocampus, to understand whether maybe the disruption of control could be linked to actually a disruption of the receptor system in the hippocampus, in the GABAergic system. Obviously, if it was the case, this will have very important consequences for potential treatment. And with this, I'm going to thank all my collaborators in Caen that have participated to the project and that I just showed you today. Thank you very much.
Somehow it's not the same without an audible clap.
I don't see any question in the chat, which it's fine for me. I'm not going to ask myself a question.
I have a question, if I may.
Yes, Maria, please.
I politely raised my hand.
Sorry, I didn't see it.
So I was wondering if you could go back to the thought, the model by Bruin you outlined initially that says you have two distinct mechanisms that could actually lead to these traumatic sort of free-floating memories, one related to overconsolidation and one related to decontextualization.
Yeah.
And I was wondering, I mean, intuitively, both could be related to the same mechanism, right? So if PTSD is related to an inability to stop replaying these intrusive memories, either online by retrieval or offline, then you would get, according to consolidation theory, you would get both overconsolidation that would go along with poor contextual integration, because you'd keep replaying them in new contexts and integrating the memories into your current context. So there would be an overconsolidation and reconsolidation, decontextualization at the same time. So can you say how if you think about these two as two distinct mechanisms and how it relates to your thoughts on where the inhibitory control mechanism might target?
Yeah. So initially, the overconsolidation hypothesis, I think, was mainly coming from study based on the fear-based learning account of PTSD. So there were a lot of work suggesting that one of the problems of PTSD that they overlearn fear basically compared to a normal individual. And the animal studies suggest that this overlearning of fear and all the study on conditional fear learning is really tightly linked to the hippocampus, to the amygdala, and not so much to the hippocampus. It's a bit linked to the hippocampus when there is some kind of context involved, but if it's fear learning based on single cue, for instance, it's mostly the amygdala that's doing the damage, basically. So this was one observation. And the other observation was that they seems also to have some kind of contextual amnesia. So it's not really clear what they mean, what it means by contextual amnesia. being that you don't have really memory for the context surrounding the trauma, for the things that happens surrounding the trauma. And this was not really well explained by the amygdala alone hypothesis. And this is why they introduce also this notion of lack of contextual integration. So I don't know how much, as you suggest, the over-consolidation can cause lack of contextual integration, but I think it was really two mechanisms that was proposed initially to account for two distinct.
Can I just build on Maria's comment a little bit? Can people with PTSD remember their flashbacks?
So there is some controversy. So because people will say that traumatic memory is not like other autobiographical memory. So there are some kind of special form of memory, and the vividness, the phenomenology of this memory is really different that you will have with other.
No, I understand all that, but I guess what I'm getting at here is, if people can remember their flashbacks, then they have episodic memory of those flashbacks. If they have episodic memory, then they have contextually integrated those flashbacks, albeit in a new context. And this is very consistent kind of with what Maria is saying, is that you're laying down a new context. I'm not sure it's consistent with what she's saying.
But that's the thing that they don't update their memory with new context.
They don't
update their memory with a new context. That's the thing, that they can't learn that the memory happens in the safe context again. That's the main problem. But
so they don't remember having flashbacks.
I think they remember the fear that they have. I think they do remember that they have flashback, but the problem that their memory, what triggers their fear, their flashback, is not updated by new context. Basically, I think it's more a form of habit memories that is automatically triggered by some cues in the environment.
Right.
And one idea that the memory system does not update the fact that they are in the safe environment and they are safe again, and that the event is not happening again.
But isn't the whole idea that what is really required to resolve the flashbacks is to reintegrate these the elements of the experience into a contextualized trace? And isn't it the case that if you have an episodic memory of a flashback, that you kind of have done that? Or what am I missing here?
Yeah, that's a good point. I see what you're mentioning. I don't know about that, but you're correct that actually point of some therapies, of some treatment, is to reintegrate and recontextualize the memory. Where inhibitions stands in this account, I think is very interesting. My non-scientific belief will be that there is a negative cycle, and that inhibition contributes to the development of the traumatic, lack of inhibition contributes to the development of the traumatic engrams, and that the stress induced by being PTSD actually cause a lot of damage in the control system as well as in the hippocampus, which exacerbates actually this inhibitory control deficit and lack of control. But clearly the point is that this inhibition deficit was not envisioned at all by model of PTSD, who only consider PTSD as a memory disorder.
You have a question, by the way.
Yes, I have one question from Elizabeth Ruiz and one question from Paolo. Hi, Paolo. What is the role of encoding the traumatic memory? In a previous talk by Gina Poe a few weeks ago, there seems to be a suggestion that preventing encoding by preventing sleep might help PTSD. I'm really not sure that preventing sleep in PTSD is a good idea. They also are PTSD because they don't sleep. Preventing the encoding of the trauma, I guess it's always a problem that it depends when you intervene. So in the early phase, some people use propranolol, for instance, to stop this consolidation process, but has to be done in some particular phases. When the memory is too much consolidated, maybe it's too late. But yes, certainly early intervention to prevent this encoding and consolidation is probably something very interesting to consider. So question from Paolo, talking about the intrusive thought, do you think hypothesize that the thalamus could play a major role? That's a great question. And Mike has some theory about the neural pathway that link the prefrontal cortex to the hippocampus. And one of these routes involves the thalamus, especially for the form of reactive control. So in the future, that would be really interesting to try to specify and highlight this route, because we haven't focused so much on this. We only focus on the functional effect of inhibition and the connection between these two systems, but we didn't focus so much on the pathway that the inhibition takes.